we commonly discuss in the class is pseudogene. So you probably already know what is pseudogene. So when we say pseudogene, we say it's a non-genic DNA fragment, which means the DNA fragment right now has no function. And usually there's no transcribed RNA, there's no protein product. Occasionally you see some pseudogene, they still have RNA transcribed, but no final protein product. So that also happened. And when we recognize a pseudogene, it's usually because the non-functionalization, when I say non-functionalization, it was a functional gene, but now lost the function in this particular lineage. So it's a non-functional copy in the organism you are studied, or in the population you are studied, or in the individuals you are studied. So it could be polymorphic as well. So the pseudogene usually is not subject to selection because a pseudogene has no function and there's no chance the gene will influence the phenotypic level. So just remind you what you learned from evolutionary biology. Natural selection acts at the individual level, and the individual has to have variation at the phenotypic level. So when we say phenotypic level, it could be morphological, physiology, like behavior, all kinds. But you have to have a phenotype. Then that phenotype will influence the mating success or reproductive success. Then that will show the differences in survival or reproduction. Then we'll say this particular trait is under selection, but the selection is at individual level. So if a gene has no function, then we say there's no chance we'll have a phenotype. But we'll talk about some exceptions later. I'll talk about how a gene lost a function but still influence at a phenotypic level. That's the special cases. Then we can discuss of that. So we say a pseudogene, it means it was a functional copy. So if it's a functional copy and now it lost a function, and we can do sequence comparison and recognize it was certain gene, say it was a hemoglobin, but now this is a non-functional copy of the hemoglobin. So we'll say that. That also implies at the sequence level, there's still a similarity with its ancestral or hypothetical ancestral copy. So we can recognize this is a particular gene, and we'll say this is so-and-so's pseudogene. So that's based on sequence similarity. And sometimes people also use the chromosome synteny and trace their evolutionary origin and to confirm this indeed a pseudogene of gene A. So that's what we did. And why in molecular evolution, people are really concerned about pseudogene, or we are interested in pseudogene, is because once the gene becomes non-functional, then we think this gene is not subject to natural selection, and we can use that as sort of like a null model to analyze the sequence variation. In other words, if there's any mutation occur at the pseudogene, it doesn't really affect anything. Because once the gene exists, especially it's fixed in the population, it doesn't really affect the fitness of the individual. And any mutation hits on the pseudogene doesn't have a phenotypic readout. So in that case, we don't really concern, we can count it as a neutral marker in molecular evolution. So that is why in the very early days, when we start have the pseudogene sequence available, we compare the sequence variation among the functional genes and pseudogenes in other region of the gene, and get some idea about the rate of change throughout the genome. When I say throughout the genome, actually in the beginning we only know a couple of genes, but now we really have the whole genome data to do the comparison. So this is like also you have learned the central dogma from DNA sequence to RNA molecule to the protein. So this is things happening like every minute in our cell. And because of that, so I just want to remind me, we have the DNA to protein. Most of the time we're concerned about the coding sequence. We're talking about the protein coding genes.